Hello, I'm Lennart Winkler, a second-year PhD student at the TU Dresden in the Applied Zoology department, and today I would like to talk about stronger net selection on males across animals. And the broader research question of my PhD could be framed as, is sexual selection an engine or a burden for adaptation? And if you look at all these sexually selected traits, you can find these ornaments, male ornament, you can find strong sexual dimorphisms, but also these weird beetle penises with spikes all over them. And you might wonder, is this not rather costly for the individuals in this population? Because these beetle penis spikes, for example, are probably not very nice for the female beetles. So overall, you could say that sexual conflict, for example, could impose major costs of sexual selection on the population fitness and also on its ability to adapt to novel environments. But there's also hope for the reputation of sexual selection, and one idea comes from the Genic capture hypothesis, stating that male reproductive success is condition dependent, and therefore sexual selection can purge deleterious mutations genome-wide. And the idea is that you have the genetic quality of an individual that influences its ability to acquire resources, and that influences the condition, and condition we define here as the total amount of resources an individual has. And this influences the sexually selected trait and the reproductive fitness. So you have sexual selection actually targeting the genetic quality and thereby purging deleterious mutations that are typically only under natural selection. And this can increase population fitness, especially if there's stronger sexual selection on males. And that is because the reproductive potential of a population is mainly limited by the productivity of females and not so much by the productivity of males. And that basically means that you have females enjoying the benefits of sharing a gene pool that is purified at the expense of stronger selection on males. And that we would term as low demographic costs. And this assumes that there's stronger net selection on males, and that is what I'm going to talk about today. And net selection we define as the genome-wide selection against deleterious alleles. But how can we measure net selection? One idea comes from Fisher's fundamental theorem of natural selection, stating that the rate of increase in fitness of any organism at any time is equal to its genetic variance in fitness at that time. And that basically means that we can use the genetic variance as a proxy for the strength of net selection, meaning that the genetic variance is a key component to estimate adaptive potential. And this means that we can measure stronger net selection in males as higher genetic variance in male reproductive success. And genetic variance you can measure as how much variation in reproductive success or the number of offspring is due to genetic components. So this is a quantitative genetic trait that you can measure, for example, by a breeding design, and this is always a population-specific measurement. And there already has been one study investigating this topic, and they looked at the phenotypic variance in reproductive success. And what they did is they compiled data from all these different studies and looked at the phenotypic variance in fitness in males and females. And what they find is that there is a significant bias towards larger phenotypic variance in males compared to females. And this indicates that there might be stronger net selection in males compared to females. But what this study does is it relies on the phenotypic gambit. So that means that you measure the phenotypic variances and you then draw conclusions on the underlying genetic variances. And that, of course, requires a correlation of phenotypic and genetic variance. And this assumes a constant environmental variance or heritability. But if there's a lot of variation in the environmental variance that you have, then the phenotypic gambit might not hold, and that is especially often true for behavioral traits, for life history traits, and also for fitness traits. And as we are dealing with a fitness trait here, it might be that the differences in the phenotypic variance in reproductive success might not translate into differences in the genetic variances in reproductive success. And that's why we have these two main hypotheses. The first is that the phenotypic gambit is not met for variance in reproductive success, meaning that we expect a weak correlation of phenotypic and genetic variance. And secondly, we hypothesize that the genetic variance for reproductive success is higher in males compared to females, indicating stronger net selection on males. And what we did is we searched Web of Science for data on the genetic variance in reproductive success in males and females, so always paired measurements within the same study. And what we ended up with is 101 different effect sizes from 26 different species. We have some mammals in here, quite some birds, one reptile, a lot of insects with many Drosophila studies, of course, one snail and one flatworm species. And first I want to talk about the phenotypic gambit now, so the correlation between phenotypic and genetic variance. And all these data points here are data points from a single study looking at the genetic and the phenotypic variance in the same design. And what you can see here on the x-axis is the coefficient of variation. That's always the measurement that I will show. So this is basically that we standardize the data by the mean to make it more comparable. And here you can see the phenotypic variance in reproductive success in males on the x-axis and the genetic variance in reproductive success in males on the y-axis. And you can see that genetic and phenotypic variance are not very much correlated. Even though there's a positive correlation coefficient, there's no significant correlation. So we cannot use the phenotypic variance and draw direct conclusions on the genetic variance. If we look at the same thing in females, we again here on the x-axis phenotypic variance in reproductive success in females and genetic variance in reproductive success on the y-axis. And you can see that also here, these two variables are not very correlated. So this highlights that it's important that we not only look at the phenotypic variance in reproductive success, but also at the genetic variance in fitness. And that's what we did next. First, I want to talk about the phenotypic variance again. On the x-axis, you can see now the phenotypic variance in reproductive success in females, and on the y-axis, the phenotypic variance in reproductive success in males. And you can see that we have divided our data into socially monogamous and polygamous species. And you can see that in orange, these socially monogamous species all fall close to this midline, meaning that there's no bias in the phenotypic variance between males and females. But if we look at the polygamous species now, you can see that most of them fall above this midline, meaning that there's a significantly larger phenotypic variance in reproductive success in males compared to females. And this is basically replicating the results of this previous study, showing that there is larger phenotypic variance in fitness in males compared to females, at least for polygamous species. And now we ask how much of this variation that we see here actually translates into genetic variance. And here you can now see the genetic variance in reproductive success in females on the x-axis and the genetic variance in fitness in males on the y-axis. And again, you can see monogamous and polygamous species. And we again find that these monogamous species all fall close to this midline, even though there's more scatter now. And that shows that there's no difference in the genetic variance in reproductive success in socially monogamous species. But if we look at the polygamous species, there again is a significant bias towards larger genetic variance in reproductive success in males compared to females, indicating that there's stronger net selection in males. And the key messages of my talk are, first, that the phenotypic gambit holds not for reproductive success, and we hypothesize that this is due to large variation in environmental variance in reproductive success. And this highlights that it's important to examine the genetic variance in fitness components as well as the phenotypic variance. And that's what we did. Next, we looked at the phenotypic and the genetic variance in reproductive success and find that it's larger in males compared to females, but only in polygamous species. That might be because there's stronger sex-specific sexual selection in polygamous species, but we have to be a bit careful here because the sample size for the socially monogamous species in our study is quite limited. But at least for polygamous species, this indicates that there could be a low demographic costs of gene capture, and thereby sexual selection can boost adaptation to novel environments. And I want to finish with thanking my colleagues, my co-authors on this paper, Tim Janicke, my PhD supervisor, Maria Moran, Ted Morrow. I want to thank my working group at the TU Dresden, the DFG for funding our research, the numerous people who provided data for this study. And finally, I want to thank you for your attention.